All right, welcome. We're going to talk about microscopes, and you are finding this in the Chapter 4 playlist because we really don't start using the microscopes until we start talking about cells. So here we go. In this video, we're going to compare light microscopes, dissecting scopes, and electron microscopes. Before we do that, let's give a pause for magnification and resolution. Magnification has to do with how many times the physical appearance of the image is actually enlarged. Resolution, however, has to do with how clear that image is and the microscope's ability to distinguish detail. If the resolution isn't very good, then the image will appear blurry. So let's take a look at a compound light microscope first. These are the ones that we have in class, and the picture you see here is very similar to the ones we'll be using in class. We're actually fortunate to have different stage clips that allow you to move the slide around the stage in a very precise and accurate manner, so we'll discuss the differences there. But a compound light microscope uses light to illuminate the specimen. The light often comes from underneath. Some also have the option of having the light come from above, but usually the light source is below the stage where the slide rests. And most of the compound light microscopes, some inexpensive ones magnify up to 1,000 times. Some of the most expensive ones can do up to 2,000 times. Compound light microscopes will not magnify more than 2,000 times because of the properties of light. At that point, bending light through the lenses just to create an image just isn't possible anymore. So magnifying something 2,000 times is about the limit on a compound light microscope. So having said that, magnifying something 2,000 times, it's good for viewing the shapes of cells, and you may be able to see a nucleus or some vacuoles or other vesicles in a larger cell, but you really can't see a lot of the organelles. So here's a picture of some yeast cells, and they've been stained purple in this case. And here is the compound light microscope looking at bacteria cells. So you see there's a huge difference in how large they are, and this is about as much detail as you can get with bacteria with a compound light microscope. And the yeast cells are larger, so again you can see some stuff, you can tell there are things inside the yeast cells, but again you're not really seeing much detail other than the shape of the cell and perhaps how they relate to one another in the overall shape. So the electron microscope, it takes electrons, and the electrons are beamed down through this long tube, and it creates a 3D image of the specimen. There are two types of electron microscopes. The scanning microscope will show you the 3D image, and whenever you see pictures from electron microscopes, they are always black and white. They're never in color, because in order for us to see color, we have to have light. And again, these are using electrons, not light. So these are quite expensive and not something that you'll come into contact at our school, at least. They can magnify things up to about depending on whether it's a scanning electron or a transmission electron microscope. Here's the magnification. The transmission microscope is called transmission because you can see all the way through whatever you're looking at. So the transmission electron microscope is what you would use to see organelles inside of a cell, while the scanning electron microscope will scan the outside surface of something so that you could tell in great detail what the outside looked like. These are great for viewing things that are smaller than cells. Here's a picture again. Here is a bacteria. You can see much more detail with the electron microscope, and again they're in black and white. You can also start to distinguish actual organelles in here, and look at the cilia on the bacteria here. Remember the little pink dot from the compound light microscope, it is now much larger and in greater detail. Finally, we have the dissecting scope, and it's often called a stereoscope, stereo meaning two because you have two eyepieces. This allows for actually viewing and manipulating your specimen in 3D. The magnification is often less than 200 times, although if you get a really expensive one, you can get probably up to about 200 times. So this is the smallest or the least powerful of all the microscopes. However, they are extremely useful. Here's an example. You could put a petri dish here with your specimen. It can be lit from the bottom. There's often a light from the top as well. You can see the light shining down here, and they're working with a small clump of dirt or something that looks like it. And they've got some tweezers. Perhaps they're trying to pick something out of that mess there. It would definitely help to have a dissecting scope. And here's one that has a movable light that you could point in a variety of directions, and again they're looking through here, and the workstation is open so that you can really get your hands in there and see what you're doing. Those are the microscopes. If you have any questions, please let me know, and that's it.